Yeah, thanks, Jess, for the introduction. As Jess mentioned, I work for Channel Islands National Park. I run the long-term Rocky intertidal monitoring program, and we also have a long-term program for the Sandy Beach community. And those programs have been going on for decades at the park. The work that I'm going to talk about today really came out of the work that I did at UCSB getting my PhD. I started this work in 2018, and it's focused mainly on something called rockweed, which I hope you'll learn a lot more about as I move through the presentation. Let's see, what else do I want to tell you just to get you started here? I think that's about it. The bulk of this project that I'm going to be talking about today was funded through the California Ocean Protection Council. And we had a bunch of collaborators that I'll introduce later in the presentation. Let's see, make sure I can advance. Okay, there we go. So throughout this presentation, just a general outline. I'll first discuss what the research problem is for this project, or was, because the project is wrapped up now. I'll give a pretty lengthy explanation as to what was the study organism, what is rockweed, why is rockweed important, and then talk about what we did to came up with as a solution for the problem that rockweed is encountering, and then provide some results from the study and then some recommendations for future restoration efforts. Well, I'm pretty sure that everybody on this call is well aware of the various threats that that we're facing for our ocean ecosystems. You know, these involve things like invasive species, of course, climate change, microplastics, overfishing, poor water quality, and then especially for the Southern California shoreline, increasing urbanization. And you know, some of these stressors are likely already causing impacts, especially to the highly dynamic margin of shoreline that is known as the Rocky intertidal zone, that's covered by high tide, by water during high tide, and then exposed to air during low tide. And just as an example, what we've noticed here at Channel Islands National Park and other intertidal monitoring programs is that the biological community has been shifting over time. And, you know, I try to explain this, and it really helps, I've found, to provide some pictures to really to show what this means. And so I picked out a couple of these pictures yesterday of a site that I know has shifted a lot. And this is a location, one of our long-term monitoring sites on Anacapa Island. And so I found this photo on the left, which is taken in 2002. And in this photo, you can see that the community is dominated by this stuff called rockweed, which I'll get into in the next couple slides, which is this olive green algae. And then the lower part of the intertidal zone has a bunch of mussels in here. And then if you look at the photo on the right, taken in 2010, and I drew that box to kind of show more of the exact area, the approximate area that's in the left. It's, I think, even to the untrained eye, you can see that it has shifted to more of a red algal dominated ecosystem. And so, you know, with community shift in mind, we recently, well, in 2021, we did an analysis that took Rocky intertidal biological community data from 74 sites along the entire west coast of North America, from sites up in, well, actually in this case, from Washington State down to San Diego. And we found that the biological community at these sites across the entire coastline was shifting it was changing during periods of abnormally warm water, such as that happens during El Niños and marine heat waves. In this case, the graph illustrates when marine heat waves and El Niños occurred around 2008 to 2009, and then again in 2014 to 15. That's the pink bars in the graph. And you see that each of these lines represent a different intertidal region. So in this case, the red line represents sites that are located along the Southern California mainland. The orange line represents sites at the Channel Islands, and green is for Central California sites. Blue is for sites that are Northern California up through Washington State. And you can see that all of these lines dip during these periods of warmer water. But what's really that stood out to us was that the southern sites below Point Conception, which is basically everything south of like Santa Barbara, have shifted the most, it seems, during these warmer periods. And the other thing that kind of stood out is that these sites try to recover back to what we're calling the initial state. In this case, is in 2007, what the community looked like. They try to rebound, but then they hit another warm water period, and it brings them further down from where they were even during the other the period before that. So along the West Coast, one of the important canopy forming algae known as rockweeds, so that I consider that just a large group of algae, they're some of the most important species within the rocky intertidal community. And we've seen that those rockweeds have shifted over time as well. In fact, they've decreased in abundance. And so you can see in this graph the cover of rockweed across multiple rocky intertidal sites in different regions. And in this case, red represents the island sites, and blue is for Central California sites, green is for the Southern California mainland sites. And they've all decreased over time since we've been monitoring rockweeds. And in this case, the Southern California sites and the island sites have decreased the most relative to Central California. And so again, I just, I like to use pictures to illustrate, like, what does this mean? How I feel like they're kind of compelling to show what a community shift really looks like, or in this case, what do rockweed declines look like? And so this is from another site on Anacapa Island. I've taken one of our fixed plots. We typically have 20 to 25 of these fixed plots at a site. I'm just showing one plot where it was very obvious that rockweed had been abundant at one point in time. In this case, from 2006 to 2008, you can see in these upper three images that olive green rockweed was fairly common in the plots, but you can see it declining in abundance and then completely disappear from all subsequent years. And I'm only showing through 2014, but it still hasn't returned. I was just there last month, and there's no rockweed anywhere to be found at this site, including all the plots. And that's the case for a number of the sites that we monitor now. And so why does it matter that rockweeds have declined in abundance? Well, largely because rockweeds are considered foundation species. So they provide a very expansive canopy cover that can be used as cover for a whole plethora of organisms when the tide goes out. And so when we have periods of offshore winds, Santa Ana winds that blow, it can get really hot and dry out there. And the the rockweed canopy can be a place for these organisms like crabs and chitons and limpets, you name it. the things that are mobile, they can get up and move underneath this stuff it retains moisture during low tide, and it stays cooler relative to outside the canopy. So in a sense, I consider rockweed to be an intertidal analog for kelp canopy in the subtidal. It provides habitat. And in addition to that, it's been determined to be highly productive relative to other types of algae in the intertidal zone, and it provides a food source, and it contributes to nutrient cycling. Here in Southern California, there are largely two species of rockweeds. We do have another species, but it's much more rare. But the dominant species of rockweed is Sylvatia compressa, and I'm not sure if you can see my cursor, but it's the thin-bladed algae that is sort of in the upper part of this photograph in the middle. And then the wider-bladed species of rockweed on the lower right-hand corner is called Pelvisiopsis californicus. And for the old timers, this was formerly known as Hesperophycus. I still call it that a lot. And generally, Pelvisiopsis occurs a little bit higher in the intertidal gradient relative to Sylvatia, but it's far less common than Sylvatia. And so for the rest of this presentation, when I refer to rockweed, I'm referring to Sylvatia. That's the species that we used for our restoration project. So a little bit about the life history about rockweed in general, but but Sylvatia, as I mentioned, is that it has a simple, almost animal-like life cycle, which is different than a lot of algae. So it makes it, it's a species that I've been studying for a long time. It makes it really easy because it consists of diploid adults that produce haploid gametes. So you have male eggs and sperm that unite to form a zygote. So it's very easy. I think everybody can get their mind around that. It exhibits external fertilization, becomes reproductive around 1.5 to 2 years old, and it can live to about 8 years. So it's a relatively long-lived alga. Eggs and sperm are synchronously released from conceptacles, which are these little pores found at the bases or at the tips of the branches that are called receptacles. They swell up during reproductive season, which is usually around beginning in November, and it occurs through spring, so March and April. But they can be reproductive almost year-round. Peak, as I mentioned, so peak gamete release occurs during daytime low tides. So it sort of evolved to drop its gametes during periods of low tide. And the thought there is that by dropping the gametes at low tide, you don't have the water movement that would take gametes and push them off into potentially other habitats that may not be preferable for survival. So the gametes drop below the canopy of the adults, which can be helpful for survival. The rate of fertilization is nearly 100%. So that's pretty amazing. But at the same time, even though fertilization rate is nearly 100%, the mortality rate of the gametes and the zygotes is extremely high, almost 100%, not quite obviously, but it's very high. So for that reason, when Sylvatia declines really significantly, it can, and especially if it becomes locally extinct, it can take a really long time for rockweed to recover or return. So let's see, what else do I want to say about here? The mortality rate that I mentioned is extremely high, mainly to desiccation, so drying out during low tide. That's a huge issue. These gametes and the zygotes can be dislodged when water comes in and it moves the adults around. It creates a whiplash effect and can push them off of the substrate. They can be grazed by a number of organisms, invertebrates, and they can simply just settle on the unsuitable substrate. The other reason why it can take a long time for rockweed to recover after it declines is that, as I mentioned, most of the fertilization and the dispersal occurs at very low distances from the parent. So since the water movement's not able to take these zygotes and gametes far distances, everything generally falls within a meter or two from the parents. So given the widespread declines that we've documented for Sylvicia, along with all these factors I mentioned that can slow the recovery of the species, we were able to make a compelling case to the California Ocean Protection Council to do some restoration for the species. And we were able to tap into a program that they call Once Through Cooling Interim Mitigation Restoration Program, which is a program they have for the power plants along the coastline that they all place money into a mitigation bank for. For this project, we worked with collaborators from UCLA. We had collaborators from UC Santa Cruz, Cal Poly Pomona, and I was at UC Santa Barbara. We had other people at UC Santa Barbara also that helped out on the project, and the Park Service was also of assistance. So an initial goal of this project was to identify the population genetics of Sylvicia at multiple locations along its geographical range. So we tried to cover just about the entire geographical range. That's one thing I didn't touch on yet. Where does Sylvicia occur? It's generally from Central California, Big Sur roughly, down south, and it goes down through Baja California to about San Quintin, roughly. But we were unable to get samples in Baja, but we did collect samples of Sylvicia at many locations where the species occurs. Using single nucleotide polymorphisms, or SNPs, we defined three main groups that are shown here in different colors. So in this case, the blue group it's the biggest group, includes sites that are north of Santa Barbara and many of the island sites. The green group includes the Southern California mainland site. So rockweed, we don't find, it's not very common north of LA along the mainland. So we have locations within LA and San Diego shown here, and those all grouped together. What really kind of surprised us is that we found a smaller group it's red here, which represents three sites on the west and south side of Santa Cruz Island. And these were all characterized by very high heterozygosity, which, compared to the other sites, this suggested a possible hybrid zone. And so these population genetics were a bit surprising to me, especially just because prior to this genetic study, we had always assumed that there was a subspecies of Sylvicia that occurred on the islands relative to the mainland sites, and that didn't show up in this study. A little surprising, and maybe more work to be done. So numerous rock intertidal locations from southern through central California, including the Channel Islands, were selected for various project components. And this included sites with healthy Sylvicia populations that served as donor sites, as well as restoration sites where Sylvicia restoration was needed. And at each of our donor sites, we got a rough estimate of the number of Sylvicia individuals that were at that location, so that we could that would assist us to determine the goal have populations that were large enough that had so that we would impact them by less than 3% by collecting for this restoration project. For the restoration sites, we identified numerous locations based on relevant criteria, which included a number of factors. The extent of the habitat, with a goal of at least 30 linear meters of habitat available for restoration, the amount of suitable substrate, so how much hard natural rock was there versus maybe boulders or sand, less suitable habitat. We needed enough habitat that was in the middle intertidal zone, of course, which is where we generally find Sylvicia. We also wanted to have some existing historical data on Sylvicia distribution, so that we knew that it occurred in this area prior, and it was in need of restoration. Other factors that went into deciding on site location included the marine protection area status. I'll point out that this project was initially focused on improving MPAs, and about a year and a half into this study, after we had already chosen all of our sites, we were told by the California Ocean Protection Council and the California Department of Fish and Wildlife that we were no longer allowed to do restoration in MPAs. This was due to an incident where, so kelp this was about the period where kelp had declined significantly along the Northern California coast and also on San Miguel Island. And there are a lot of proposals were being submitted to the California Department of Wildlife to do restoration in kelp forests, including removing urchins, and these were proposed for MPAs. And it turned out that the language just was not clearly written about MPAs and what you can and cannot do in them. And so it was determined to pause any restoration work in MPAs at that time. And I think it's still largely the case. So we had to pivot and move our sites outside of MPAs. So let's see. We also I'll just finish this slide by saying that we chose a total of 12 locations, with 7 sites for restoration were chosen in Southern California, in either Palos Verdes or San Diego. We chose 3 locations on Santa Cruz Island to represent the Channel Islands, and then 2 sites were chosen north of Point Conception. We used UAVs to map out changes in rockweed abundance over time, and we use them to produce 3D digital elevation models for each of our restoration sites. And so we operated the UAVs at low elevation, which allowed us to acquire surprisingly high resolution data. You can make out some of the rockweed transplants in the image on the right. There are these brown clumps, which I found pretty surprising. I think that's a really useful tool, and we're slowly integrating UAVs into our program, our long-term monitoring program, because they're just so useful. Prior to doing any of the restoration work, we set up plots within the Sylvicia zone at each of the sites, and we assessed the biodiversity of sessile and mobile species. So sessile species meaning most of the algae, some of the things that don't move around as much, limpets, and then the mobile species like crabs and mainly crabs. We also established transects along the intertidal gradient, which allowed us to measure changes in biodiversity as a result of restoring Sylvicia at a site. And we repeated those surveys several times during the study period and then again at the end. So, so in an effort to restore the Sylvicia, we identified the healthy Sylvicia populations, as I mentioned, for donor sites, and we extracted fertile adult plants. So I'll mention later in the presentation that I did some work for my master's that explored different factors that affect restoration success for rockweed, and we found that adults were had higher success as survival. So we focused on adults for this restoration project, and also focused on fertile adults, so that if we, the hope was if we move those individuals into a restoration site, that they might drop gametes and produce some recruits while they're there. So here you can see that we carefully removed each of the rockweed individuals with just simple hand tools. I had originally started using angle grinders and tools that will die if they hit salt water, and we found that actually if you have the right substrate that breaks easily, then a chisel and a hammer work just fine. And the idea here was to chip out enough rock underneath the holdfast, which is the disc that the rockweed uses to attach, so that you can then epoxy that whole piece into your new site without affecting the holdfast ability to hold onto the rock. We found that if you get epoxy onto the rockweed, that could impair its ability to survive. We collected a total of 2,110 Sylvicia plants, and we outplanted them into 12 different locations. So after collecting the rockweed, we then transported them in coolers, and we moved them to the recipient sites. In the case of the sites that I worked at, which was on Santa Cruz Island, there were a lot of logistics involved in doing that, because we collected rockweed on one side of the island, then we had to move to the other side, and we had to use a boat to access the sites. And so you can see that image on the left of us moving the coolers onto the site. We then wrapped each of the individual Sylvicias, thalli, and saran wrap in an effort to keep the epoxy off of them. We used the epoxy to glue the Sylvicia thalli in place into plots that we identified in the along the proper intertidal gradient, and we transplanted between 50 to 150 Sylvicia thalli with the rock attached to them into each of our restoration sites. So in some cases, multiple restoration efforts were conducted within a site over time, and this sometimes resulted in several hundred transplants at a single site. I should note that we attempted to place Sylvicia transplants in a similar tidal range as those observed at the donor sites. For the monitoring, we set up restoration plots at most of the restoration sites within zones where natural rockweeds were present, or at the tidal levels where Sylvicia should be present, because at some of our sites we did not have any Sylvicia left to give us an indication of where to put our restoration plots. But we, in those cases, used proxy species to determine the right zone. So, for example, there's a red alga called endocladia that is commonly in the same intertidal gradient as Sylvicia, and so that was generally present. So we would use that to help us identify the tidal range to put our plots. We marked each of those plots with stainless steel hex bolts in each corner so that we could relocate them. And we transplanted 4 Sylvicia individuals in each, generally in a single clump within each of those plots. And we found that by clumping the individuals together, that enhanced survival as opposed to just having them spread out. So within the plots, we monitored survival and the growth of transplanted rockweeds, the recruitment of new rockweeds. We also sampled the community composition. And in addition, control plots were established in natural rockweed beds within or near the restoration site when possible. The transplants within the plots were monitored to measure survival, initially on a monthly basis, and then we did it every 3 to 6 months after the first year. We monitored for approximately 2 years. We got a late start on this project. It was a 5-year project. This happened right during the height of COVID. And then we had that MPA issue that I mentioned. So we we lost a little time for monitoring. Let's see. The survival of individuals within natural Sylvicia control plots was also monitored over the same period. And every 3 to 6 months, a subset of the recipient sites, so the restoration sites, we measured the growth and the reproductive potential of Sylvicia that were translocated into the plot. So essentially, we're looking at the size of the plants and then also the relative number of receptacles at the tips of the branches, which hold the reproductive material. Let's see, how am I doing on time? Okay. So since the methodology that we were using had a small but still a negative impact on donor populations, we tested a variety of novel restoration methods. So one of the first things that we looked at wasn't necessarily to increase. It was more of to increase efficiency. We were looking at whether or not we could just paste or glue down the holdfast directly to the rock, which would really speed up operations. But we kind of knew that wouldn't be very successful, and we found out that indeed that does not work. We had to continue gluing down the rock and keeping the epoxy off of the rockweed plants. But then we start looking at other methods of doing restoration. So one method involved manipulating the release of the gametes from receptacles that we collected from plants without taking the entire plant. We just took tips of the branches off and we induced the release of those gametes, which is a fairly simple thing to do even in the field. And then we then poured that inoculated solution of gametes onto a variety of surfaces, both natural surfaces like just rock, as well as a type of red algae that I mentioned, Endocladia, which you can see in the image on the bottom left, is that red algae. We find that Sylvicia recruits to this algae quite easily. So we tried to pour the solution over the Endocladia in hopes that we would get recruits that way. We also tried pouring this solution over an artificial substrate, which is shown here as that green scrubby pad in the right photo, that we hoped would mimic the same thing as Endocladia, the red algae. And the gametes would get stuck in there and hopefully recruit. Let's see. Anything else I want to say? We also tested the effect of tidal height on transplants at a subset of sites to determine whether there were different patterns for survival based on what tidal height individuals were translocated within. So we put transplants above and below what we determined to be the Sylvicia zone. So moving into some of the key results, because we did a lot of stuff, I'll just hit on the highlights here. Overall, we found that the survival of transplants was highly variable across restoration sites over the course of the 2-year project. And in this case, the number of outplanted individuals remaining at sites ranged from just a few transplants to approximately 75 at the most. What this equated to about 0 to 45% survival after 1 year. And then after 2 years, that survival dropped down to 0 to 38%. I admit the font on this graph is not the greatest, not the easiest to read, but basically what you want to take away from here is that percent survival is on the vertical axis, time is on the X, the horizontal axis, and each of these little tiny dots represents a different site. And in this case, this red line and red shaded area represents a 95% confidence interval for the trend of restored plots, the plots where we put transplants into. The blue represents plots where we were following the survival of plants that naturally occurred. And so you can see that there's quite a discrepancy between the transplants and the controls. Recruitment of Sylvatia was also highly variable among sites, as well as over time. However, in this case, the temporal and the spatial patterns were similar in restored plots and the natural plots. And again, the red represents the restored plots and blue represents the individuals in controlled plots, so the naturally existing Sylvatia. And when there's overlap, that shows that the level of recruitment in this case was approximately the same. And you can see the photos on the right just kind of give you a better idea of what we're looking for when we're looking at these plots. So in this case, the orange arrows in the orange box represent the location of new recruits. And then, the white arrows in the white box show where we put our transplants within these particular fixed plots. Perhaps not too surprising to us, at least, that given the very short period that we had to do monitoring after we did the restoration, the community structure within the intertidal zone as a whole did not differ, both if looking at before and after doing the restoration. And this suggested that the Sylvatia restoration had no impact on the community structure of both algae and invertebrates. Despite the relatively large number of Sylvatia individuals that we collected within each of the donor sites, there was not a significant difference observed in the magnitude of percent change over time. So this indicated that we did not have any detectable impact of our collection on the donor sites. Overall, our restoration techniques required relatively low levels of effort. It was a little bit more challenging working on an island, but for the most part, it was fairly efficient. We were able to move or collect 30 individual Sylvatia plants per person per hour. And we could transplant slightly fewer, so 10 individuals, Sylvatia individuals per person per hour. So let's see the takeaway here. Overall, our restoration objective was to establish Sylvatia populations with an adequate number of individuals surviving over time, along with the evidence of new individuals recruiting and surviving over time. As we expected prior to starting this work, the true assessments of the success of our restoration efforts are likely to take several years, beyond just the 2 years that we did monitoring. But based on the patterns that we've seen so far, we believe that restoration was successful at 4 of our sites, with one of the sites actually showing evidence of a self-sustaining population. And this is much sooner than we expected. But at the same time, the success of restoration at these 4 sites requires more time to monitor. And unfortunately, we had another 4 sites that were considered complete failures. We lost essentially every transplant that we put into them. We did expect some failure in the beginning, but it was definitely surprising to us to have complete failure at a site. And it's unclear right now the reasons why those failures occurred, and it's likely attributed different factors for different sites. But we did find some geographical patterns with 3 of the 5 locations that are including the Santa Cruz sites and then the Central California sites. 3 of those sites completely failed. Whereas like the sites on the Southern California mainland all fared fairly well. None of them completely failed. One of them was not great. We had very low survival, but we did have high survival in some of the other sites in Southern California. So this project provided several lessons that we should consider to maximize future restoration success. And one of them being that we recommend you conduct Sylvicia restoration at multiple sites, since restoration success can be highly variable spatially, and it can be challenging to identify which sites will result in success a priori. And we found that even sites with existing natural populations of Sylvicia have the potential for restoration to fail. Secondly, we recommend conducting Sylvicia restoration over multiple years. This is because if the initial restoration at a particular important site fails, success can be highly variable temporally. So basically saying that abiotic conditions during some years are better than others. Number 3, be prepared to abandon some sites. If you're seeing high mortality of outplants, we found that it's, even though, because we did try to redo our restoration at a number of sites where we saw failure the first year, and we continue to get failure at those sites for one reason or another. So be prepared to leave them. Our 4th recommendation is to assess the success of restoration over several years following transplantation, since it could take a long time to see whether or not it was successful. In some cases, it can take 5 to 10 years. And finally, continue to more robustly test restoration approaches, because it's not clear that we've found the best method yet. I think I would like to end this talk with just a ray of hope, just because we did, I think had hopes that this project would have been a bit more successful than what we found. And we really don't have many proven tools to improve the condition of rock intertidal habitats. And this is probably in a large part due just to the challenges involved in working in such a complex environment. But rockweed restoration does have the potential to enhance the entire rock intertidal community in terms of increasing biodiversity and resilience. And that's because rockweed is a foundation species. So as I mentioned earlier, I did some work for my master's project working with the same species. I looked at, in a scientific manner, looked at various methods of restoring that species, as well as factors that could affect the success of survival. And in doing so, I only put out a very small number of transplants, around 100 or slightly more. But when the project was all over, I had less than 70 transplants still present at that site, which was in Little Corona del Mar and Newport Beach. And for many years, there were really no changes that we could observe in that tiny population of Sylvicia. But it did persist. But we were excited to see that more than a decade later, so in 2022, you're looking at a photograph of that site where rockweed had just all of a sudden kind of boomed. And now there are contiguous areas of rockweed bed throughout the site. And so I kind of like to just leave it at this, and hopes that some of these locations where we did some restoration will potentially look like this in the future. And so I think that's about what I have. I would like to acknowledge all of these people that helped out on this project in various ways. Some of them were field assistants, others were PIs. It was a fairly big effort. And so I do want to acknowledge everybody's assistance here. I also would like to point you towards this story map feature that was done on the project called Racing Tides. This can be found on the Southern California Research Learning Center website for the National Park Service, but you can also just take your phone and take an image of this code here on the upper right-hand corner, and it'll take you to that link. And I think that's all I have. So hopefully we have a few minutes for questions, and I'll go ahead and stop sharing.